So last time we had finished talking about the fecal oral route and how the parasites mostly affect the GI tract, right? And so we're going to continue on with that discussion today. And so if you think about the parasites, remember there are three different groups. There are the protozoans, which we are talking about now. They are the metazoans. Metazoans is a word that just simply means they're multicellular. And then there are the exoparasites, and we're going to cover those today. And then that's all I have planned today. I know that I'm behind, but I'm doing my best to try to catch up without overwhelming um, people with information. Okay, so there are six groups we're going to talk about, and we begin with the Archaeazoa. So remember, the Archaeazoa are those organisms that are transitional organisms between bacteria and true eukaryotic organisms, right? They have a nucleus, right? So that would define them as a um, as a eukaryote, but they don't have mitochondria, and so therefore they have their electron transport system in their plasma membrane, much like the bacteria do, right? And so there's some really important pathogens in this group, right? The two that I am going to talk about are Giardia lamblia and Trichomonas vaginalis, right? So this is what Giardia lamblia looks like. As you can see, this is a trophozoite. Remember, the trophozoite is the active free swimming state of the protozoan. And so this is what it looks like. Those are not eyes, those are sucking discs, right? So they're not eyes, right? And this is what the cyst looks like, right? It's oval in shape, and there's usually about 4 nuclei inside of it. Okay, and then this is Trichomonas vaginalis. It is the etiology, but cause. Etiology is cause, right, of a sexually transmitted infection that we call trichomoniasis, right, or just simply trick, right? And so if we look at these organisms, right, we'll start with Giardia. So there's a person that looks kind of like me doing exactly what I would do out in the wilderness and loving nature, except one thing that he's doing that I would never do is if I see these little critters swimming around in a pond, these beavers, I am not going to drink from that water because the beavers, along with a lot of other mammals, are the natural reservoir for a lot of parasites, including Giardia lamblia, right? You get infected with Giardia lamblia, you will have a very malodorous type of diarrhea, right? And I've had it because mostly the way I got it was I drank out of my well on my property, right? So I found out I had a well, I opened it up, pumped out some water, and drank it. I tested it for bacterial pathogens first, and there were none. So I said, okay, that's good. So I drank it, and then I got a case of giardiasis, right? Which wasn't a lot of fun, but it's easily treated. It's easily treated, right? And so here you can see the organism that this is an electron micrograph of the organism, and so you can see the organism, right? And it's very small. It's um, the, the type. It's just slightly bigger than a bacterium, if you will. But you can see that nutria and beaver and a bunch of other organisms, right, are the natural reservoirs. The disease in some states is known as beaver fever because of the beavers actually being kind of the reservoirs of it, but there are other reservoirs. Deer can be a reservoir, right? And so it's an important thing to understand. Okay. Any questions about Giardia and the fact that it's an Archaeozoan? Okay. Trichomonas is on the rise. It is a sexually transmitted infection. The interesting thing about Trichomonas is that it doesn't have a protective state. So it is only trophozoitic. It only has an active free swimming state. So it goes from the penis to the vagina and the vagina to the penis and back and forth, right? Because it doesn't really need any protection. It's not going to go through any types of. Um, so the life cycle that you'll need to know is malaria, James, but you'll need to know you'll need to know in general, you'll need to know the trophozoite and the cyst and what they function as, okay? But then when we get to talk about malaria, you'll need to know the entire life cycle for malaria, okay?
I'm sorry, you said the cyst and what? When
The
did
trophozoa,
we? I don't remember covering that. I'm sorry.
yeah, you're probably getting coffee. Right, right there. Okay, that was last time. Okay, James. All right. So if we think about this, because this organism goes from the penis to the vagina, it doesn't need any protective state, because it goes from one nice inhabitable environment to another, right? It doesn't go through any types of stomach acids or anything like that. And so it doesn't really need protective. So it has evolved so that it doesn't really have a cyst. It only has a trophozoitic state. It only has an infective state, if you will. Okay. And so this particular organism has an axostyle. The axostyle is the way that it attaches to the tissues, and you can see it right here. It's this appendage right here. Okay. And so it causes what we call strawberry cervix in women, and I'll show you this later on when we talk about mechanisms of infection in Unit 5. Okay, but for right now, you just need to know that Trichomonas vaginalis is an archaeozoan, and it causes a sexually transmitted infection. Okay. The second group are the Euglenozoa. The Euglenozoa are also flagellated, just like the Archaeazoans, but they have not only a nucleus, they have mitochondria. And the two organisms that I'm going to talk about here are those that are going to be hemoflagellates. They're going to cause infections in the blood, right? And so they both belong to the Trypanosoma species. One of them causes African sleeping sickness, and the other one, closer to home, in the Americas, causes Chagas disease. Okay, so this is what they look like, and if you were to stretch them out, they would be about the size of 3 erythrocytes, right? So a little bit bigger protozoan, right? But so you could actually see them in blood. They're fairly easily discernible in blood. Okay. Trypanosoma that causes African sleeping sickness, right? There are several different species of them, but you can see that the organism is transmitted to humans by the bite of a tsetse fly, right? And so this is what the tsetse fly looks like. It takes a blood meal from you. It injects the organism into our bloodstream, and then over a period of months to years, depending on our physiology and our immune system, the organism takes hold and causes problems. It becomes neuropathic, right? It affects the nervous system. And eventually what happens is the nervous system just kind of shuts down, and people pass away very quietly usually, right? It's not like malaria where, my God, you go out with a bang, right? So this one, you pass away very, very quietly. Okay. Trypanosoma cruzi, closer to home, right? It's been in South America, Central America, and in Mexico for many years. It is now in the United States on the southern borders, right? And we have seen cases of Chagas disease in Arizona. We have seen some in Texas now. And some researchers out of Texas went down to South Texas and captured a whole bunch of the regiviid organism, this particular organism, also known as the kissing bug, right? They captured a lot of that the regiviid, right, organism, and you spell that. Regiviid, right? They captured a whole bunch of these, and 60% of them were positive for Trypanosoma cruzi, the etiology, the causative agent of Chagas disease. Okay, So if you take a look at the diversity of these organisms, right? So remember, the protozoans are classified by how they move, right? So far we've just talked about the organisms that move by flagella, right? But they're both transmitted by different vectors, right? Where the tsetse fly transmits the organism that causes African sleeping sickness, and the rejuveate bug, or the, if you will, the kissing bug, transmits Trypanosoma cruzi, which causes Chagas disease. Okay, so with Chagas disease, it gets into the bloodstream, and then eventually it can cause cardiomyopathy, right? It can cause heart problems, but it can also cause problems within the intestinal area, right? It can interfere with the blood supply, cause um, abnormal problems with the digestive system, and if not treated right, the chronic disease can lead to death. This is in Texas, along some other states, right? This is going to be our new big problem. It's already a problem here. People just don't know it yet, because I mean, there have been some articles and some news pieces about it, but people just don't know how ubiquitous this particular infectious agent is in Texas right now. And really, we see more cases in dogs than we do with humans. But I just think that the organism is um, is thriving in the human population, and we just haven't seen kind of the effects of that organism getting into the human population yet, right? One of the things that early on is a symptom of having Chagas disease is this type of neuropathy that's called Bell's palsy. And with Bell's palsy, you can see that half of the face gets temporarily paralyzed. Now, Bell's palsy is a symptom for a lot of different things, right? Lyme disease, a stroke, um, um, MS, multiple sclerosis. There's a bunch of them, right? And so you just have to be sure that you're looking for all the possible causes of what the what the Bell's palsy might be giving us a clue for what's happening to our patients. Okay. Yes, Barbara.
Do they like do the how do they determine what it is? Like, is there a blood sample they can take, like a long?
Uh-huh. Yep. They can do that. There are really fast tests nowadays to look for the organism in the blood. But for the other ones, right, for if you're trying to rule out some of the other things, there are other diagnostic tests you can do for MS and strokes and a bunch of other things too. So there is quite a few protocols to be able to do this. But if you're just looking for the infectious agent, right, then there's some really quick tests you can do, and I say quick, a day, right? Takes about a day, but you can take a little bit of blood, and then um, you take a little bit of blood, and then you can run the test. Lucia, so for everybody, it's going to be a little bit different, right? Some people aren't going to show symptoms for years, right? Some people may show symptoms in months, right? But I think in the United States, especially, right? If a bug comes into the house, there is an all-out war to destroy that bug. Are you with me? So I don't think we're going to have too much problems with people kind of in the house, right? But individuals who like to be outdoors a lot, who like me, I like to take a nap in my hammock in the backyard, right? These organisms, the, the the kissing bugs will land on your body, and then they will take a blood meal in a vascular part of your body, right? So typically, the most vascular part of the body that's going to be exposed to these organisms is the face. And so they'll take a blood meal from the face. You won't even feel them. They're very, very stealthy. Um, and when they take a blood meal, they also defecate, and the organisms, the the trypanosoma, are released in the feces of the organisms, and then they get into the bloodstream by getting into the portal, the opening where the organism took the blood meal, right? So it's almost adding insult to injury, right? Where they take a blood meal, they cause a hole in you, a small hole, and then the organism, the one that's going to cause you real problems, gets in that way, right? Yeah, pretty interesting life cycle. Questions? Okay. So a pretty interesting life cycle. You do not need to know this life cycle. I am just showing you the life cycles because I want you to understand that almost all these organisms have developed a very complicated life cycle, right? The two parts of the life cycle that you just need to know for now are the cyst and the trophozoite and their functions. But when we get to malaria, you'll need to know the entire life cycle, and I'll explain that to you when we get there. Okay? All right. The amoebozoa. So these move by pseudopoda, right? And so I'm going to show you this particular slide just because I want you to be aware of what these pseudopoda look like, right? So here is the cell itself, right? This is the cell, and then you can see the pseudopoda, and the organisms move toward that pseudopoda, right? It's very dynamic to watch these organisms and how they move. They're pretty beautiful organisms. Most of the amoeba that are out there are not going to be problematic to us. They're not going to cause any problems to us, right? But there are a couple that can cause really serious damage. One of them is called Entamoeba histolytica. And this is the one that you can get from different places, right? Where they don't have really good infrastructure to make sure that everything is taken care of hygienically. And so you can pick this up usually from contaminated water, but also, food. And these organisms get into our body through the fecal oral route, right? So you eat your food or you drink your whatever, and then the cysts get into the stomach. The stomach weakens the cysts. The cysts then go into the small intestines and they break apart, and the active free swimming state of the protozoan gets in, and it causes problems for us. Okay? Yeah. Dysentery, amoebic dysentery is what it causes. All right. A lot of people say if you freeze stuff, right, then you're going to kill the organisms. That's not true. So if you're out and about in different countries, you just need to be real. You need to be real mindful of what you're doing, right? Just because you're drinking a frozen margarita doesn't mean that the freezing the ice and the alcohol and the Everclear and the or the you know the alcohol in the margarita is not going to kill the dormant state, the protective state of the protozoan, the cyst. It's going to get into your body real quickly, right? And whether or not it develops depends on a lot of factors, but if it gets in, then it has a good chance. The other organism are the ones that we typically see as associated with causing infections of the eyes and the brain, right? These belong to the Acanthamoeba group. The Acanthamoeba and Naegleria, right? Those two amoeba, you get into your body because you're just splashing around in the lake or whatever, or you're jumping off into the lake or the river off of a zip line or a rope that's tied to a tree, right, or whatever. And when you jump in, the water goes up your nares, goes up your nose and back into your sinuses, and then it can get back into your brain, right? This case with this particular eye right here, we had an individual who had contacts in and jumped into the water, and the amoeba got underneath her contacts, and she didn't take her contacts out. She left them in overnight, and then her eyes were irritated the next morning and didn't think too much about it, right? But then tried to remove her contacts, and there was a lot of damage. That it is very painful. Not only that, James and Mercy, but it caused blindness in this particular person, right? So the Provi's rules of water, right? Not published anywhere, just a little bit of advice. Number one, if the water is stagnant, don't get in it, right? Stagnant means it's not moving, right? Number two, if you've had a rainfall, right? Give the lake or the river a day to clear up and then get in it. Don't get in it right after a big rainfall, right? There are thousands of things in there that can make you sick because of everything, as we talked about when we talked about nutrition and growth and the population curve. When you have that rain, everything washes into those bodies of water, right? Give that body of water a little bit of time for its dynamic systems to clean it up and the sediment to fall to the bottom or to be carried away. Okay. And then the last rule of water is if the water is very warm, right? Not nobody's gonna carry a thermometer around to measure the temperature of the water. But if you get in, it's very warm, right? 80 degrees Fahrenheit or warmer. Go someplace else, right? So these kiddos are playing in this water, but that water is really warm, right? And so the amoeba really like warm water, right? And fresh water. The amoeba do not do well in salt water, so you don't have to worry about these if you're going to the beach or the coast, right? You don't have to worry about them. The amoeba are not going to be there because they just can't handle the salinity, right? Their cells will burst, so they don't do well. But they do really well in fresh water and in us also. Okay. Any questions about the amoeba? So the things you need to take away here are that they're classified in the way they move, and they move by pseudopoda, right? And then a couple of them, one of them causes dysentery, and the other one can cause infections of the eye and the brain because they get into our body through our nostrils, and then they get in that way and cause all kinds of problems. Okay. The Celophora, an interesting and a beautiful group of organisms, but not very many pathogens at all in the group, right? As a matter of fact, most of these organisms, most of the Celophora, are in symbiotic relationships with other organisms. So did you all know the termites can ingest wood, but they do not have the enzyme systems to digest the wood, right? So the termites will eat the wood, but then the bacteria and the protozoans that are in the intestinal tract of the termites will digest it, and then all the organisms, including the termite, benefit from that. So that's the type of symbiotic relationship you're going to see from this. One of the organisms called Balantidium coli, see how beautiful they are here, look, just really beautiful, not really dangerous usually. One of them, Balantidium coli, is a known opportunistic pathogen to cause a form of dysentery, right? Causes a form of diarrhea, right? It has a life cycle that most people would consider complicated, but mainly because it has more than one step, right? And so you can see the cyst gets into the body, gets into the stomach, gets weakened, gets into the intestines, right? Becomes a trophozoite, becomes an infective state, causes problems, and then some of them form cysts and are released through the feces, right? Again, you do not need to know the life cycle, right? I'm just showing you because one of the general themes of the parasites is that they have all developed a very complicated life cycle. And that complicated life cycle gives them a really good chance to survive in a competitive environment, right? Us or nature, right? Because if you break one part of the life cycle but do not affect the other part of the life cycle, it just comes right back. Okay. All right. The last group of true protozoal parasites are the apicomplexa, and these are the bad boys of the group, right? They are horrific. They cause horrific diseases. I'm going to talk about three of them, but there are a lot more, right? And so if we think about these guys, they are all going to be intracellular pathogens, right? They don't move, right? So they're not motile, but they have this little kind of battering ram that's associated with them, right? And we call that an apical complex, right? This is known as a ring structure, right? It is the trophozoite of malaria, right? So you can see that this particular red blood cell is infected with two infective states of malaria. That malaria parasite will reproduce in the red blood cell. The red blood cell will break apart, and all of these infective states will be released into the environment and infect other red blood cells. And so that's what causes the signs and the symptoms of malaria, which is horrific: high fever, bed shaking chills, right? It's It's just a terrible, terrible disease infection. If you want to see a really good movie about malaria, the movie's name is Mary and Martha. It's about two mothers who lost their children. One of them was an adult, the other one was a little kid. But it's probably on Netflix or Amazon Prime or whatever. And so you can watch it if you have time, right? So maybe sometime after the summer. Okay. So these are all of the different stages of the malaria life cycle. We're not going to do all of these, right? I've broken it down to some real simple categories so we can talk about them, and we'll do that right before we start talking about the metazoans today. Okay. But the organisms that I'm going to talk about are Plasmodium falciparum, right? That's malaria. Plasmodium falciparum. I'm going to talk about Toxoplasma gondii, right? You've heard of that one. It's usually carried around by cats, and then Cryptosporidium parvum is the other one, and that causes a form of diarrhea, dysentery, especially in immunocompromised individuals, and we'll talk about those, right? We'll start with malaria. So here's a really great image that was taken. It's a scanning electron micrograph showing the malaria parasite coming out of the red blood cells that they have infected. You can see it's been color enhanced so that it sells magazines, but it really is a really beautiful micrograph showing that. This is the organism that transmits malaria, right? It doesn't cause malaria. Malaria is not caused by a mosquito. The mosquito is what we call a vector, right? Just like some of those other organisms we were talking about earlier, right? The tsetse fly and the regiviate bug. The mosquito's a vector, right? In science, in biology, in microbiology, a vector is an organism that transmits an infectious agent from one organism to another. Okay, so this is a big time problem. It's epidemic, and but unfortunately, not every continent has malaria, right? So Asia has malaria, Africa has malaria, South America has malaria. But the European nations, most of them do not have malaria, and North America does not have malaria. So you can see that we cannot call it a pandemic, although it kills a lot of people on an annual basis, right? On an annual basis, we lose almost the same amount of people to malaria as we've lost to COVID currently, right? Yeah. But nobody's talking about it. Why are we not talking about this big problem? Well, because the countries that have the means, the resources to help with the problem of malaria do not really care about it because it doesn't affect the citizens of the countries that have those resources, right? Americans only want to talk about what bothers Americans. Are you with me? We don't want to talk about what bothers other people. That's just the way we are. So if malaria is not here, we're not worried about it, right? We're worried about Zika, right? Oh my God, there's Zika here. Oh my God, I gotta do something, right? Well, Zika doesn't kill near as many people, not even a fraction of a percent as many people as malaria does. But we are not talking about malaria because it's just not affecting us, right? Good. There are some Americans that go over and go on vacation or on business trips to other countries and come back with malaria. That's possible, right? But they're very few. And then we do have individuals who decide to come here from other countries and live here and become citizens of the United States, and they have malaria, right? So we have those. But other than that, malaria is just not here. It used to be here. It used to be in Florida and Mississippi, Alabama, and Louisiana, and in the Houston area of Texas. But we did such a good job of getting rid of the Anopheles mosquito that malaria just simply didn't survive, right? We attacked the life cycle where it was most vulnerable, right? Where the mosquito needed to be used in order to get the malaria parasite into the human, and we took care of the mosquito problem. And then once that happened, we didn't have malaria anymore. As a kid growing up, in the late 70s, early 80s, I can remember a truck coming by and sticking a hose in the sewer system and just letting rip all this huge plume of mosquitocide, right? Of an insecticide that would kill mosquitoes, right? And we almost never saw mosquitoes because of that, right? But malaria is a problem, just not a problem in the United States. That doesn't mean it's not going to ever be a problem here, but it could be. We're going to talk about the life cycle a little bit later, right? I've got it set up so that we'll talk about it, but we're going to get through all the protozoans and then we'll talk about the life cycle. So Toxoplasma gondii is the other one. Again, it's a bigger problem in people who are immunocompromised than it is in pregnant women, but anything that affects babies is going to be a big deal. Are you with me? So therefore, Toxoplasma gondii gets a lot of attention from causing problems in pregnant females, although it's so rare that, you might have to, I don't know, I don't even remember the statistics because it's just so rare, but it can happen, right? So Toxoplasma gondii is typically found associated with mice and rats, and it emboldens those mice and rats so that they challenge the predators of it, right? And so, like for instance, a mouse sees a cat, it's emboldened to go over and fight it. That's because the parasite itself changes the physiology of the mouse or the rat. And now that mouse and the rat challenges the cat kills it, eats it, and now it becomes the reservoir for Toxoplasma gondii, right? And its dormant state, its cyst, we call it an oocyst, is released through its feces, right? Therefore, if you are a pregnant female, you should never clean out the kitty litter box, right? Let the other person in the relationship that isn't doing anything for the next 9 months do that work. But be sure you train him, because guys think that cleaning out the litter box just simply means yeah. Guys just think that cleaning out the litter box is taking the litter box outside and throwing it on the lawn and giving the lawn a little bit of extra fertilizer. That's not true, right? You don't want to have the organism around in the very rare case that it could cause an infection in the female. What you do with the litter box is you scoop up the waste and the litter and put it in a bag, no dust, a plastic trash bag. Yeah, yeah. Well, uh, right, but that's the way you should do it, right? You should just simply clean out the litter box and then throw that away as regular waste in the trash can to go to landfill. Do not put it in your garden, right? Yeah, yeah. If yeah, yeah. Well, you'll be surprised, James. I grew up with a bunch of crazy boys. Yeah. Yeah. Oh, no. We used to have a cat. I don't have any. Miss Kitty passed away, but I would clean out a litter box, and I didn't wear a mask, but I'd put it in the regular trash, right? But if a pregnant female was to contract an infection by Toxoplasma, that could cause an abortion of the developing fetus, right? Yeah, that is true. But think about this for just a minute. Just think about this for just a minute. It causes much more problems in people who are immunocompromised than pregnant females, right? Yeah, pregnant females are a little bit immunocompromised for sure, but I'm talking about our AIDS patients, our cancer patients, our myasthenia gravis patients, things like that. Right? What I will tell you, James and Lucia, is that if we were to take the entire population of ACC and everybody would agree to donate just one tube, 6 milliliters of blood, And we could test them against all these infectious agents that we're talking about, or we will talk about. About 85 to 90% of us would test positive for being exposed to, that means have had Toxoplasma in our bodies, because it's so ubiquitous. It's everywhere, right? It's with cats, it's with birds. Both of those, cats and birds, are the natural reservoirs, but there's a lot of others, right? And so I test positive, right? When you're a lab person and you're working the late shift at night and you're running all these tests and there's extra reagent that's going to be thrown away, why not test your own blood and see what you test for? So we would do that a lot. And you know, I test positive for this organism, just like a lot of other people would, right? Because it's everywhere. Okay? Questions. Very complicated life cycle, right? So you can see the mice, right? The cats eat the mice, or come in contact with other animals that can be reservoirs, and then the cat and the human come in contact with each other, or the human can contract it from other animals by eating their meat, and the meat isn't cooked properly, and then you can have problems, right? So it's a pretty complicated life cycle. All right. Cryptosporidium parvum. This is a problem, and we do have outbreaks of this organism every once in a while. As a matter of fact, we have had an outbreak of this organism. It causes a form of diarrhea. And we've had an outbreak in Cedar Park, and we've had an outbreak in San Antonio. Those are the two places closest to us that have had outbreaks of cryptosporidiosis, right? Organism that gets into our bodies through the fecal oral route. And a lot of us are going to test, I test positive for, a lot of us are going to test positive for this organism because we've come in contact with it. We've been exposed to it. But we don't get very sick from it. Are you with me? But our AIDS patients, if they contract this organism, they will have diarrhea for almost the rest of their life, right? Therefore, if you remember when we talked about antibiotics, I said that there are a few antibiotics that cross over a little bit and can be used prophylactically to make sure that certain organisms don't show up in our immunocompromised patients. And a Bactrim sulfa drug can be used to make sure that cryptosporidiosis doesn't show up in those AIDS patients, if you will. Okay. A great movie to watch if you haven't watched that makes reference to cryptosporidiosis is God, it's been a while now. It had Tom Hanks and Denzel Washington. And Tom Hanks was a lawyer, but he contracted AIDS. It's Philadelphia. Thank you, James. Thanks. Yeah, great movie. It's just been a while since I've thought about it. But that particular movie, toward the end of the character that was being played by Tom Hanks, toward the end of his life, his best friend, he gets up and goes to the bathroom, and she tells Tom Hanks' lover, who was played by Antonio Banderas, and also Denzel Washington, that he goes to the bathroom about every 20 to 30 minutes, because what they're making reference to is he's infected with cryptosporidiosis. Okay. It is a very dangerous organism to certain segments of the population, the immunocompromised patients. To the immunocompetent, to the person who has a really great immune system, not going to be a problem. You might get infected, but it's not going to be a problem. But to somebody who doesn't have an immune system, or their immune system is compromised, it is a problem. Okay. These are both electron micrographs. This is the cyst, and then here you can see the trophozoites coming out of the cyst. This little area is called the apical complex, and that's how it gets into the cells that it infects. Okay? Questions? All right. Complicated life cycle. I don't even have to know it. The last group of organisms that we'll talk about in the protozoal group, but this is not a parasite, right? This doesn't get into our body and cause disease that way. It causes toxins, right? Are the dinoflagellates, also known as the Pyrrophyta or the fire algae, right? These organisms are known to cause red tides and brown tides, right? And they're problems for not only us, but they're problems for lots of other creatures and nature, right? So here you can see how they are related phylogenetically, right? They are considered to be a part of the apicomyplexan tree, right? So they're related to malaria, if you will, but they are algae, right? And so they are planktonic. They are in the oceans of the water for the most part, and they cause red tides, but really it's the toxins that they release that are the problem, right? And these toxins can be really bad. They are also, they also bioilluminate, right? So I think I told you that I love Puerto Rico. I've been there almost, I don't know, 9 or 10 times, something like that. But on the Caribbean side, we'll get in, we, we've gotten in there at night and we just kind of start splashing around at each other. And the dinoflagellates all bioluminesce, and it's really beautiful to watch, right? It's almost this kind of bluish green bioluminescence, right? But the thing is that you can see other organisms in the water, right? So one time we did that and we saw a shark and we got out of the water. But the organisms are beautiful. It's only when there's an increase in nutrients, right? If there's an increase in nutrients because of a lot of rain, because of fertilizer, because there was a hurricane, because there was a flood, all this water washes into the estuaries and the localized areas around the seashore, right? And so this is off the coast of Louisiana, and this area is all hypoxic. What that means is there's no oxygen. It's a dead zone. Nothing lives there anymore, right? And so that's just because there's no dissolved oxygen there anymore. And that's mostly because of the pollution that's occurred in that area, right? Well, this area will give rise to these other areas that are nurseries for other marine life, and then that's what can be affected by the tides, right? So you can see kind of the green tide that's shown up here, but over here in the bay there's no problem, right? Because there's a barrier, right? And so you can see the algal bloom here, right? Remember, what causes that to happen is there's all these nutrients and the algae just go crazy and they start to grow out of control. This is a red tide. Karenia is one of the organisms that will cause a red tide. There's others, but this one is a very common one. And the red tides, not only is there no oxygen in that water, but there's a lot of toxins also. And the toxins will affect just about every single type of life. It will kill everything, right? This happened in Corpus Christi in the early 80s when I was growing up there. And I can remember all these fish washing up on the seashore, and people were picking up and eating them. And I didn't know anything about micro, back when I was, an 8th grader or 7th grader or whatever. But it was an amazing thing to see all these people. And the health department of Corpus Christi was saying, do not pick those fish up, right? Because potentially there could be this toxin that we call Saxitoxin. Saxitoxin, and it's a neurotoxin, right? And it biomagnifies in things like fish and shellfish, oysters, right? So when I was growing up as a kid, I'd go fishing all the time, and my mom or my dad would drop me off at the jetties, and I'd go out there and I would fish for about 8 hours, and then I would call them and they'd come pick me up, or I'd walk home. But my lunch, usually I had a lemon and some salt, and I would pick the oysters off of the jetties and I would eat the raw oysters for lunch. That was my lunch. It was delicious. I won't do that anymore. Almost all the bays in Texas are too polluted for me, right? But the oysters that come out of the North Atlantic are just delicious. And so if I know where they're coming from, I will eat them raw, but usually I don't anymore. I will cook them in roux or fry them up or saute them, but I'm gonna cook these things because the cooking will destroy the toxin itself. Okay? This is why these organisms are important. I think you might remember that about 4 years ago, there was a hurricane that came across. Yep, they sure are, because they're filter feeders. Yeah, for sure. That's a good point, James. That's a really good point. What do you mean? Abdullah, what do you mean decaying fish? Like rotting fish? Okay, go ahead, James. Okay, thank you, James. That's what I would have said. Yeah. Uh, Abdullah, I don't. Can you can you provide some content around that statement? Why are you asking? Um, oh yeah. Okay, so if it's fermented, ceviche or what, if it's that kind of stuff, yeah, that's okay. If it's cooked in acids, oh no, no. Okay, so let me clarify. I said that there were people in Corpus that were picking up those fish, not me. All right, I was like, I looked at my dad. I said, "What the hell are they doing, Dad?" And he said, "I don't know. I should go check it out." My dad was always like on the lookout for any kind of deal he could get. So if the fish were going to be good, he was going to go pick them up. But we never did. Yeah. Okay. I'm sorry. I'm sorry if I made that statement that no, I would not eat rotting fish. No, absolutely not. Okay. Or as James said, it's a hard no. Yeah. Good. All right. So this is what the toxin looks like. You do not need to know the molecular structure of a toxin, but I need you to know it's a neurotoxin. And what it does is it causes a paralysis, right? The syndrome itself is called paralytic shellfish poisoning, right? It's much like, James, I don't know if you've dealt with this. It's much like the tetrodotoxin that you would find in the blowfish or the fugu fish. James, any experience with that type of culinary? Okay. So as a matter of fact, I want you all to know that the zombie lore um, is really about viruses infecting people and causing them to become zombies, right? But it didn't start out that way. Zombies were produced only by voodoo practitioners who were using tetrodotoxin or saxitoxin to put people in a stasis, right? To put them in a in a deep, if you will, coma, right? And so the zombies started out that way, right? If you want to watch a movie about it, Rainbow and the Serpent. And then it became where, so they moved real slow, right? Dawn of the Dead, right? Or Night of the Living Dead, that kind of stuff. And then all of a sudden, I was surprised when Walking Dead came out and the dang zombies were moving faster. And then when World War Z came out and they started to run, I'm like, hell, we ain't got a chance now. But the zombie lore has continued to evolve with our need for being afraid of things, right? And so it's pretty cool to think about. All right, so if we think about the dinoflagellates, right, Karenia, right, it causes red tides and brown tides, which the organisms, they will produce toxins, saxitoxin, and the saxitoxin is a neurotoxin, and it will basically shut down our nervous system, and we die, really, because we stop breathing. Okay? Questions? All right. All right, we're going to talk about the life cycle of malaria, right? This is what you'll need to know. And I don't like memorization, but you can memorize this one because this is what I'll ask about, right? So the life cycle is in two parts. There's a sexual and an asexual part. The asexual part happens in the humans, and the sexual part happens in a mosquito, right? And so it's a cycle. It can start anywhere. And so we're just going to start right here. We're just going to start. I need a different color. How about red? How about, let me see if this works. Okay, good. We're just going to start right here. Okay, so a mosquito takes a blood meal. And the mosquito that takes a blood meal is the vector for malaria. So it has malaria in its salivary glands, right? So when it takes a blood meal, it injects a little bit of an anticoagulant, right? Does everybody know what an anticoagulant is? What is an anticoagulant? What is an anticoagulant? Somebody tell me. Stops blood from clotting. Because when the mosquito takes a blood meal, the blood, they don't want the blood to clot at their proboscis as they're taking a blood meal. So they inject a little bit of it's like heparin, if you will, blood thinner. And so that will allow the blood to flow freely from the human to the mosquito. Are you with me? But in that salivary gland, and where that anticoagulant is, that's where the malaria parasite is. So it takes a blood meal, it injects the sporozoites, right? The, if you will, the diploid version of the organism, the sexual cycle, right, into the blood. And once in the blood, it migrates to the liver, our liver, and it matures, right? It differentiates, it matures, and it becomes a trophozoite. The trophozoite in the malaria life cycle is also known as the merozoite, right? So let's be sure we all know that. The trophozoite is exactly equal to the merozoite. Okay? They're equal. Okay. So once these things get released from the liver, they will infect red blood cells, and they continue to do that over and over again, right? So this is a continuous cycle, right? Where the merozoites infect red blood cells, they produce more merozoites, and this just continues on. And that's what causes the signs and the symptoms of malaria in us, okay? Eventually, some of those will differentiate into male and female gametocytes, right? So these are your haploid cells, if you will, right? And another mosquito takes a blood meal, and when it takes a blood meal, it has to ingest both a male and a female gametocyte. They migrate to the lumen, the intestinal tract, small intestines, if you will, of the mosquito, and they conjugate. They form a zygote. They come together, right? And when they form sex cells, right? The sex cells, again, are going to be your sporozoites. They mature. The sporozoite breaks apart, or the zygote breaks apart, releases the sporozoites, and the sporozoites migrate to the liver. Migrate to the salivary glands of the mosquito. And then the mosquito another mosquito takes a blood meal. It injects the sporozoites into another person, and the malaria life cycle continues on and on and on. Okay? So really all you need is one individual to come to the United States with malaria, and you need the Anopheles mosquito, and we can start our own outbreak of malaria in the United States, especially around the areas of the United States that are more subtropical, right? The areas where malaria once was, right? Florida, Mississippi, Alabama, Louisiana, Texas, right? It can happen, right? All we need is a starter. Okay? Questions. Now, this particular life cycle is known as an alternation of generations, right? Because each generation produces the next. So the sexual cycle produces the asexual cycle, and the asexual cycle produces the sexual cycle, right? And it continues on like that forever and ever until we somehow or another attack. The way to get rid of malaria would be to attack the malaria life cycle in multiple places. You cannot just cure the patient. The patient gets cured, fine, but you cannot just get rid of the mosquito, right? Yep. Alternation of generations. An alternation of generations. Okay. Good. So, which brings up sickle cell. So, somebody tell me what sickle cell is. What is sickle cell disease? Somebody tell me.
It is when the red blood cells aren't formed completely into the disc. It has like a sickle shape.
Right. And so the malaria parasite does not recognize that as being infectable. As a matter of fact, people who just carry the trait have resistance to malaria. So what we can say is, and this has been proven, is that the malaria parasite cause an adaptation in humans, especially around the areas where malaria is found normally. And that adaptation was sickle cell disease, because people who have sickle cell disease or who carry the trait don't get malaria. The problem is what? The problem is that sickle cell is worse than malaria, right? So evolution doesn't get it right all the time, right? And so you can see then that we have this huge problem with malaria. So the way to get rid of malaria is to attack it and destroy parts of the entire life cycle, right? Not just one. You just can't treat the patient, right? You have to attack it on multiple. And if you do that, we can get rid of malaria. We have the technology to get rid of malaria, right? But we don't. Yes, Justin?
I feel like I've heard this before, but the patient could have sickle cell trait, but not actually have sickle cell
That's
anemia.
that is true. It can be a carrier of the trait, right? It can have the genes in the DNA, but the genes don't get expressed, right? And that just means that they would be heterozygous carrier, right? And so if we think about sickle cell as being little s because it's recessive, and then the person who has it would be big S. If a person is big S, little s, they're carrier, right? But they're not gonna show the trait. But in that case, you're right, Justin. You are absolutely correct. That they will not. They are resistant to contracting malaria, but they don't have the sickle cell shaped red blood cells. Oh, James put it much more eloquently than I did, right? He said that you have to get the recessive gene from each parent. That's correct. Mm-hmm. Okay. Questions? Any questions? Yeah, yeah, yeah. So it's a really good thing to talk about when you're talking about circulatory problems and things like that. Uh-huh. Yeah. So it is a beautiful mechanism. It's a beautiful adaptation, but evolution got it wrong, right? Because people with sickle cell. It used to be, now James and Justin, it used to be that there was almost no treatment, right? When I was growing up, I had a really good friend who had sickle cell. And in 10th grade, he told us that he was not going to get any more transfusions. What that really meant was he was going to die. He and his parents talked it over, and he just said, I don't want to live like this anymore. His parents kept telling him, in the future there might be a way to treat this, and it'll be better. And he just said he was tired of it, so he stopped getting treatments and he passed away that year. But that was his choice, and his parents were allowed him to do that, right? But nowadays, there are treatments for it, and with CRISPR, I think I told you that there have been at least two cases where scientists have gone in and edited the genes from people who had sickle cell and have cured two individuals from sickle cell by taking out the genes of sickle cell and putting in normal genes that are normal for the normal blood cells, and it's completely cured these two individuals. There were four people who they did this to. Two of them worked, one of them didn't, and the other one they're still trying to figure out what's going on. So that provides a little bit of potential way to cure sickle cell in the future. I think that's pretty exciting. Okay. Questions, anybody? So James, yeah, because that's where the stem cells are. Yeah, so that's where you'd have to do that. Mm-hmm. Okay. All right. Let's go on and talk about Abdullah. There is a ton of research going on with CRISPR right now for all kinds of things, cancer, all kinds of really great things, right? So yeah. So I did want to show you the difference between normal red blood cells and sickle cell traits before we went and talked about the metazoans. And then one of the reasons that, or two of the reasons that sickle cell disease is so problematic is, of course, the sickle-shaped red blood cells do not carry oxygen very effectively, nor CO2 away from the body. And then the other thing is there are clots that are formed in the smaller vessels of the body because of the sickle-shaped red blood cells who form these aggregates and then have problems, right? So it's a terrible infection. Okay. All right. Let's go on. Let's talk about the big guys, right? So we're going to talk about the tapeworms. We're going to talk about the roundworms. We're going to talk about the flukes, and then we're going to talk about the exoparasites. So just so that we are aware of time, I have about 20 minutes, and then I'm not going to go any further than that because we still have a lot to do, and I want to give time to talk about the review for the practical. So wherever I get in 20 minutes, that's where we'll stop, and we'll just take it up next Wednesday. Okay? Any questions? All right. So if we think about these larger organisms, right, especially the things that look like worms, right, we're going to talk about those guys first. So they belong to the kingdom Animalia, and they are the platyhelminths, which are not true worms. These are going to be your flukes, which we call Trematoda, and they're going to be your tapeworms, which we call Cestoda. The other organism, the other group, are the Nemahelminths, right? The Nemahelminths are the true worms, and they're going to be your roundworms. And so those are the only real worms. Everything else looks wormy, and people call them worms, but they're not really worms, right? They're all chemoheterotrophs. They have to consume something else in order to gain their nutrients. There are none of them that are photosynthetic, right? They have reduced digestive system. They have reduced nervous systems, and they can move. And they move by either wiggling around, or they have grooves, right? And so they can move, right? They're ingestive and they are absorptive. They have very elaborate life cycles. So playing on that particular theme of having these real complicated life cycles. The difference here is that all of these organisms form embryos, right? They all have sex and they form embryos, right? So we're having a lot of these organisms being formed. All right. So if we think about that for just a minute, right? Here are the different things, right? So these are the trematodes, these are the flukes right here, right? And so they're flat. They don't have any segments, but they're multicellular, right? These are the cestodes right here. These are the tapeworms. And so you can see that they have segments. And then underneath this image that got moved when I moved it into Blackboard are the roundworms, right? The nematodes. And these guys have grooves, right? And the grooves help them move around, right? So if we think about how the metazoans are classified, they're really classified by the three S's, right? The three S's, if you will, right? And those S's are shape, sex, and segments. The three S's, okay? That doesn't mean that they all have segments. That just means that the segments play a role because, I just said the nematodes have groups and they kind of look like segments, but they're not, right? The shape is very, very distinct. And sex, it's not about do they have sex, they have sex, but it's about how their structure is. Do they self-fertilize or do they have to undergo conjugation with another organism? That's a different. In other words, do they have both sex organs or do they have two different genders, two different sexes, if you will? Okay, that's how we classify them. Okay, so remember we talked about the most important parts of the life cycle in protozoans. Somebody tell me what those are. What's the most important parts of the protozoal life cycle? Cyst is one of them. What's the other one? That crazy word starts with a T. What is it? Trophozoite. That's good. In the metazoans, it's the ova and the adult. Okay. So yeah, these things can get really big. Some of these organisms that I have seen, right, are up to 80 or 100 feet long. They're enormous. They're large, right? They're pretty cool, but they are large. The ova act as a fingerprint, because they all look different, just like the fungal spores, right? You can see that general theme over and over again. Oh, how do you identify? Well, you identify them by their asexual or their sexual structures. And here again, we identify them by their structures. Now, in the fungi, it's mostly their asexual structures, but in the metazoans, it's their sexual structures. It's their ova. Okay. All right. So there are two terms, monaceous and dioecious. Monaceous, this is how they have sex, right? Monaceous means that you have an organism that has both the male and the female reproductive organs in them, right? So here is a fluke, right? This is a liver fluke. This area here is the testes, right? Here's the seminal vesicle. Here's the ovary. And this, ladies and gentlemen, is the uterus with the eggs in it. There's a little pore right here, and that little pore is where the eggs come out and get released into the environment. Okay? So this is an organism that is monaceous. One organism, mono, aceous, meaning sex, right? And so, what do you mean it starts with? I don't. What do you mean it starts with? So, I'm not sure. No. So it would. Oh yeah. But that's the fertilized egg. So let me clarify. So it really starts with the production of the sperm and the testes, right? And this production of the egg and the ovary, and then they get fertilized, and then they get put into the uterus. Okay, Barbara? It's just kind of like us, also, right? Um, the ova and females get produced in the ovary, they get released into the fallopian, they go down the fallopian tubes, but in the fallopian tubes, a sperm hits them, it gets fertilized and eventually ends up in the uterus and attaches to the uterine wall. It's kind of like that, right? It's just a lot smaller organism. Okay? Questions? The other term is dioecious. And dioecious means you have two distinct genders, or you have a male and a female. Are you with me? So here is Ascaris lumbocoides. The male has this little hook end, right? The female doesn't have a little hook end, but in the worm world, typically the female is much larger than the male because the female is gravid. What does that term mean? What does that term mean? They're gravid. No, no, not muscular. That gravid means that they are with egg. They're with progeny. All right. So we wouldn't say that to one of our friends. Oh, you're gravid. No, we wouldn't say that. We would say, oh, I'm so happy you're going to have a child. Right. That's what we would say. Right. But it's the same thing. Okay. They're with egg, or they're with progeny. That's what that means. Okay. So everybody understand the difference between monaceous and dioecious. Okay. So here's the rule. Write this down. Trematodes and cestodes are mostly monaceous, right? There's a couple of exceptions, okay? And the nematodes are dioecious, okay? Good. Trematodes and cestodes are mostly monaceous. There's a couple of exceptions, but the nematodes are all dioecious, okay? Any questions? All right, let's go and talk about these things. So again, here is the liver fluke, right? Clonorchus sinensis, and we're going to talk about it in just a minute. So the trematodes are the flukes, right? Ever heard that term? It must have been a fluke, right? So your kiddo won a game and they're not on a very good team, and you said it must have been a fluke. It all derives from the fact that a parasite is a fluke. You can get a fluke, but it's very rare, right? So it came from that terminology. These are flatworms, right? And they're flat because they really are flat. I mean, if you lay them down, they are flat, right? They're just one, look like a pancake if you want, right? But they're small. So here's Chlamorchus sinensis. It is a liver fluke. It's when it attacks the fluke, it's very painful. So here is a transverse section through a liver, and you can see when they cut through the liver, there is 1, 2, 3, 4, 5 flukes in this small place, right? And you guys who took anatomy and physiology know that the body is quite wonderful. If it can't get rid of something, what it normally does is it sequesters the problem, right? Therefore, you have sesamoid bone, or you have overcalcification of the bones when they get broken. In the lungs, when you have tuberculosis, you have calcifications of the lungs, also in the liver, right? So these calcifications, these caliculi form around the flukes, and it sequesters them so they don't do as much damage, but the damage is being done anyway, right? So you can see these calcifications in the liver. Okay, good. All right, so that's a pretty small one, right? So you can see the naked eye, but if you're not looking, you're not going to know it's there, right? But you can see it with the naked eye. You cannot see the internal structures with the naked eye, but you could see it with, say, 100x objective. Pretty easy to see. Okay, so they're pretty big. Right? Very complicated life cycle, right? You don't have to know any of these life cycles, but you do need to know that in the metazoan life cycles, the ova and the adult are the most important. Okay, so you can see here there is a primary host, a secondary host, and then the final host, right? And we as humans do not have to be the final host. We're kind of an accidental final host. There's a lot of other organisms that eat raw fish that contract these things. Okay. All right. Here is Schistosoma mansoni. So I told you that the trematodes are mostly monoecious. This one is one of the exceptions, and so I wanted to show it to you. This is a blood fluke, right? So you can see here, and this particular organism breaks all the rules of the worm world. So what did I say about male and female? Which one is usually bigger? Which one is usually bigger? Correct, female. This one breaks that rule. Are you with me? Here is the male, right? Kind of a cute little guy. There's the male, right? And the female is right there, right? She has to crawl inside of him in order to undergo sex, right? So here you have 3 different images showing the conjugation matrix of the male and the female. Are you with me? Yeah. It's a pretty cool organism. Now this one is a bad boy, right? You don't want to contract this. We don't have these in the United States, so you don't have to worry about them. But other parts of the world, they can get into the body because there is a larval form of it called the Cercaria that actually penetrate the skin when people are wading into the creeks or rivers or ponds or where they're growing rice or whatever, and then these organisms get in them, right? And so it can be pretty devastating with these things all over your blood. Pretty bad. Okay. Yeah, that's correct. So eventually, yeah, usually, so when these Cercaria get in, Mercy, the sex hasn't been appreciated quite yet, right? You don't know if it's male or female, but eventually when they get in, they'll be male and female. Some of them will be male, some of them will be female. Okay. Questions? All right. Here is a very large organism. This one is so big, Fasciola hepatica, it will fit in the palm of your hand, right? So if you have 2 or 3 of these guys, it's taking up a nice big volume, a nice big portion of the liver, right? Here you can see the calcifications in the liver, right? But these things are enormous. They're huge, right? The ones that I have in the lab are under, they're permanent slides under, so you can look under a microscope. And so they're probably about, I don't know, 3 centimeters, right? But they get much bigger than that. Okay. So these are trematodes, and that one is Fasciola hepatica. Let's talk about tapeworms, right? So tapeworms can infect every single part of the body, but we're normally going to see them in the small intestines, right? There are some parts I need you to know. I need you to know the head. The head is called the scolex. Be sure you know it, right? And I need you to know the different segments of the body. The different segments of the body are known as proglottids. Okay, that's the term proglottid. And each of those proglottids are going to have both the male and the female sex organs in them. So you can see them right here, right? The testes are here, and there's the ovary, and the ovary produces ova. They get fertilized by the sperm, and out they come through the genital pore, and they get released into the environment. Okay, the environment could be our own small intestines, and some of these could then grow into mature tapeworms, or they can be released through the feces and contaminate the environment, or contaminate your fingers, and then you shake hands with Justin, and then Justin has a sandwich, and then Justin has a tapeworm issue, right? So some of you may have heard from other family members, Mercy, you must have a tapeworm. You're so skinny, right? And so that's because, tapeworms are known to take away nutrients from the individuals that are infested or infected with them, and they lose a lot of weight, right? Angeles, what do you mean where they're found? Everywhere. In Austin, Texas. They're found everywhere. Okay. So mostly they're going to be associated with livestock, right? But other animals will have them also, and that's how we acquire them. We acquire them through the fecal oral route. Okay. Now let's get back to the scolex. So what did I say the scolex was? What did I say the scolex was? The head. So the head can either have suckers, all right, or it can have hooks and suckers, right? And so it's pretty cool to think about how these organisms have evolved, right? So if they have hooks and suckers, they're going to be much more readily attached to you, and they're going to be hard to get rid of. If they just have suckers, they will use their suckers to attach to you. But either way, these things are easy to get rid of. All we have to do is take an anti-helminthic drug, and it paralyzes them, and out they come. Right? They'll come right out of your intestines and into your toilet, right? Yeah, sometimes piece of them break off, and then you get to see pieces of them in your toilet, and you're like, oh my God. Yeah, and those are the proglottids. Okay. My wife and I moved to Austin, and we were living by Riverside on Parker Lane. Our next door neighbors knocked on our door at 3 o'clock in the morning, and she said, I know you know about these things. Can you come over? My son has got worms. And I looked in there, and she said, it's tapeworms. I'm like, it's not tapeworms. It's a pinworm, which I'll talk about in a minute. But yeah, they look like little flakes of tissue, right? I'll show you what they look like in a minute from an image from cat poo. Okay. So here, back in the day when I first started teaching at ACC as an adjunct, I would ask my students to be on the lookout for a freshly killed possum or raccoon. And they would call me up and say, Prove, there's a dead possum at the corner of Slaughter and Manchac. So I'd go down there, pick it up, I'd bring it to the lab, and we would dissect it in the lab as part of our course, right? I can't do that anymore. ACC doesn't like me to do that, so I can't do it anymore. But I used to have a permit; we could do that. I would never take I would never take an animal's life, but if there was a fresh kill, we would do that. And possums and raccoons are notorious for having all kinds of parasites. So this is an image that was some of the ones that we took when we were in class. And this is not that image. I just took this out of the web, but this is like them. And so you can see, I don't know, there are about 15 tapeworms in there. But one particular possum that we dissected one time had over 60 different parasites in it: protozoans, nematodes, tapeworms. It was crazy, right? Yeah. Oh, so don't be afraid of them. Although, James, you got to be careful with those coons because they hold grudges, right? But yeah. So yeah, I once threw a rock at a coon, and it challenged me. It came at me. So yeah, Yeah, and so this is the time of year for them to be having their brood, right? Yeah, yeah. So it's pretty crazy. Here is what we Tania saginata. This is the beef tapeworm. It only has suckers, and here you can see the tapeworm coming out of animal's anus. Are you with me? So I had the same problem when my dog was alive. My wife took her for a walk, and my wife called me from the park. The park is like 4 houses down. She said, "Come over to the park now. Bandy's got worms." And so I ran over there, and sure enough, my dog was looking a little bit concerned. Why is everybody looking at me? And out from the back of her anus was this long, elongated structure coming out of her anus. And I looked at my wife, and she looked at me. She said, "You're the microbiologist." And I said, "Oh yeah." So I just grabbed it and started pulling it. I knew what it was already, and I just pulled it until it released. What Bandy had done, my dog, is we had the Kleenex that has lanolin in it, right? And the dogs like the way that oil tastes, and so she ate quite a bit of box of Kleenex. We just got lucky it didn't cause impaction. That it didn't obstruct her intestines because we would have been having surgery. But it rolled up nicely and it came out of her anus. And so for a couple of days there, I was picking things out of her anus, right? But she didn't have a problem after that, right? So you just have to be careful. So if you have tissue around and it's got lanolin, you've got a dog, just keep it away from the dog. Put it up a little higher, right? This is Taenia solium. This is the pork tapeworm, right? Pigs get a really bad rap, mostly because they'll eat anything, right, including you. If you are decaying and dead and the pigs run across you, they're going to eat you for lunch. Are you with me? They're not picky. So that's why a lot of people say they feed pigs slop, right? It's whatever, right? When I was working at a grocery store going through college, undergrad, um, I was good friends with the guy who worked in the produce area, and he would just throw all kinds of rotted or decaying fruits and vegetables into a big old bin, and then a local rancher would come over and pick it up and feed it to his pigs because they don't care. They eat everything, right? But here you can see that this organism, where Taenia solium only had suckers, right? I'm sorry, Taenia saginata only had suckers. Taenia solium has hooks and suckers. You can see the hooks right there. Okay, complicated life cycle. This is the bad boy of the group, Echinococcus granulosum. This particular tapeworm is very small, right? It has a proglotid and it has a scolex and 3 proglottids, right? It produces these egg sacs, right? So you can see this particular necropsy of this animal has these egg sacs, and inside of them are the immature form of the larvae, which we call the cysticercus, right? So this is problematic because you can see that there are other egg sacs that you might not be able to tell if you're just being really hone in on these, right? There's others in here, right? You can see them forming. And so they're all throughout the body, including in the muscle, right? So this is a big problem, right? And so with these particular types of tapeworms, during the first recordings and reporting of the AIDS outbreak in San Francisco and Los Angeles in the early 80s, some of the epidemiologists and the doctors that were working with the patients were finding that this particular organism was causing infections of their brains and other parts of the body. So they didn't know what to do. So they called up some large animal vets and said, "Well, what do you do?" And the large animal, "Oh, you mean when we have cattle that have this?" And they said, "Yeah, we just shoot them, right? Well, we're not going to shoot humans, right? That's just not going to happen." But you can see this can be really, really problematic very quickly. And so the only way to remove these things is to surgically go in there and remove them. And so if you've got one egg sac, that's easily removed. But if it's in the brain, it is a problem. So this really necessitates surgical intervention, right? Okay. So I said I would stop at 12 o'clock. It's now 12:01, and we're going to stop here today. We're going to, well, I want to show you, here's cat poo, and you can see the proglottids in the cat poo, right? But other than that, we'll start on next Wednesday after the practical. We will start with the nematodes, and then we'll finish that, and we'll talk about viruses, and hopefully finish viruses on that day. Is there any questions about anything we talked about in lecture?